Okay, so let's start the meeting. So Suresh, Natalie, and Maimuna, can you hear me? Okay, let's start. Okay, so hello everyone. Thanks for joining the SNEV EV Characterization Series. My name is Amir Nasiri. I'm a postdoctoral researcher at the University of Tokyo and also the former president of a student network on EVs. So I'll be moderating today's session along with my colleague, Natalie, who is the SNEP executive member and PhD student at the Queensland University of Technology. So the title of today's talk is Vesiclepedia ExoCarta FunRich, Bioinformatics Tools for EV Research. So it's my great pleasure to introduce today's speaker, Professor Suresh Mathivanan. So Professor Suresh Mathivanan undertook a PhD in proteomics and bioinformatics at the Institute of Bioinformatics, India, and Johns Hopkins University in US. After PhD, Suresh joined Ludwig Institute for Cancer Research in Melbourne, Australia, as a postdoctoral researcher. In 2011, he received an NHMRC Peter Doherty Fellowship to study extracellular vesicles secreted by cancer cells. Suresh moved to the Department of Biochemistry at La Trobe Institute for Molecular Sciences at La Trobe University after receiving a LIMS fellowship to set up his own research group in 2011. At LIMS, Mathivanan's laboratory is focused on extracellular vesicles, their role in cancer and intercellular communication. He has authored over 118 articles that are cited more than 35,600 times. His lab has contributed key studies in biochemical characterization of EVs as well as the bioinformatics capability. As example, ExoCarta, Vesiclepedia, and FunRich. So thank you so much, Suresh, for accepting our invitation. It's really our great pleasure to have you in our SNEP EV characterization series. So I'll hand it now to you. So please start the presentation. Thank you.
Thanks, Samir, and thank you, the SNEV committee, including Natalia and Maimuna. So thank you for your patience in putting up with me, and then thanks for the invite. So I will probably start this off with a bit more journey into the EV research as well, like how I got into the EV field per se. But at this moment, again, this is a different kind of a talk that I'm used to because normally, again, we don't, I never present or talk about Vesiclepedia ExoCarta or FunRich in any of the conferences or where I go, because where we do in terms of cancer research and EVs on that. So that's where I focus most of the time. But in a way, in this particular session, so I started my postdoc in Australia in 2008 under Professor Richard Simpson. Again, some of you might have heard of him, some of you might have known him. So he's a well-known person in proteomics. And when I finished my PhD in proteomics and I'm looking for a postdoctoral position, somehow I was looking in terms of countries where they speak English, and I was looking at Australia. Even though I did my PhD in US, I thought I'll come over and see how the life in Australia is. So at that time, Richard had published one paper on exosomes already. And then when I joined the lab, he introduced me to exosomes, and he showed this paper from Jan Lotwall, which was published in Nature Cell Biology. Again, all of you might have known about the shuttle RNA. And then when I read the paper, I was kind of very attracted to the concept of cells sticking to the same place, but they release these vesicles where they can have proteins and nucleic acids, and these guys can travel within an organism, and even cross-species communication is validated nowadays, and then elicit a lot of function on them. So I started working on that. And at that time, I was working on colorectal cancer cell-derived EVs, right, exosomes. So when I first characterize them, and then I'm looking at what are the proteins which are commonly found in EVs, and these are general questions which we ask even now. And is there a common signature among various cell types, common signature irrespective of the source? Then I started looking at previous studies that has been published on EVs. there were not a lot of proteomic studies published at that time. There was one study on urine, and then one on bladder cancer, if I remember. So then I compiled a list of those EV proteins and then compared it against what was identified in our study. So then I realized that if I'm doing this, everyone else in the world is doing this. So why can't we create a compendium? Right? So what is present in EVs from all the sources? And then while I was doing this, because I also had an education after my master's, I did a bioinformatics course. I can program myself. I started say I can create this resource, and I was able to do that in quick time. In a way, I created this database, online database, using Python as a programming language. Then I used Zope for a content management system and MySQL in the backend. So I can pretty much upload the data into there and then create this. So what I did is I manually went to all the articles at that time. Luckily, there was less than 75 articles at the time, but not anymore. As you already see, we cannot cope up with this literature on EVs anymore. It's too many. Again, we only go to a particular theme and we're interested in now because it's too many articles published nowadays. So at that time, 75 was what we had. So I started compiling them in terms of experiments as well. So me being the only curator at that time, so I could control a lot of those things easily, and the quality control I could measure really well, and then exactly I know what to do. And I was trying to capture a lot of metadata in terms of how was the EVs isolated, what rotors were used, and things like that, to an extent, not comprehensively at the earlier stages. And then I called them exosomes at that time, right? But again, I'm sure a lot of your audience, I don't want you to jump on me at the end saying that these are not exosomes. Again, we all know at this juncture that what people called as exosomes may not be the bona fide exosomes, because we do know that a lot of heterogeneity is there within the vesicle subtype. So though we created this exosomes compendium, slowly interest started building on other vesicle types as well, right? As we all know. So this was published initially in 2009. As you can see, this was the initial publication in Proteomics when we described this. And I thought, okay, this is probably the last of it, and we're not going to go into other EV subtypes mainly. But then when we started to understand this more and more, we understand it like the complexity in terms of the biogenesis. And then we also started appreciating there's more EV subtypes than what we know of. So last, when we updated this ExoCarta, we had somewhere like 286 studies, and these are the statistics that you can see, where we had around 42,000 protein entries. So what do we have? How do we have this ExoCarta? Like at the moment is, if an author kind of says this is exosome, and this is from a small EV pellet and 100K, we kind of restrict those kind of studies only to ExoCarta at the moment. But given that we know more, we have plans in terms of going back to ExoCarta and then trying to keep only very confident small extracellular vesicle studies in ExoCarta, and then remove some of the other ones which we think is not in the small EV compartment, right? That's our future goal. But at the moment, it has data from small EV and 100K pellet, which also has a lot of confounding factors that. But however, even if we do go in that path where we are restricting this to only certain EV type, that is SEV alone, we have to accept and understand the fact that this is not still going to be an EV subtype with homogeneity. So the cargo and the content that we are reporting there, it is going to come from a lot of other EV subtypes as well. But however, we are going to base this on studies that have enriched for the small EV, so we could have the likelihood that the content that we are getting is most often found in small EVs than in other EV subtypes. But however, no one can be sure until they validate in their experiments. So what do we have here? Again, we have this cargo which has been incorporated into EVs, and that is cataloged here. So if you go into the download, anyone can download the data. It is not restricted only to academics. Company people also can download it and use it for free. And you will have the meta annotations as well, and you can restrict a study based on, like, say, you're only interested in, like, say, OptiPrep or a density gradient centrifugation based methodology, and then you can look at the studies which have been reported in that. So this is a snapshot of how it looks like. Like if you go there and you can browse ExoCarta, then you can get a molecule page. And when you go into a molecule, this is ALIX. Again, ALIX being one of the escort component, accessory component, and very important in the biogenesis of EVs. You can look at it and then see in the metadata section, you can have the experimental ID. You have reports in terms of, let's say, you have mass spectrometry based identification for ALIX and EVs, and that has been denoted here. You click on that, you can get the MSMS spectra for that and look at the quality of the spectra. This is not available for all the data, only for those who are able to provide the MS data can be accommodated at this particular time. Then you also have other meta-annotations, most importantly, the sample source type, and as well as what is the isolation methodology that was used to isolate this particular EV subtype. Then you also have the interacting partners of ALIX, and those which are in purple, again, these are all the proteins which are also identified in EVs, and those which are in blue are those which are not, or as of now, not detected in EVs per se. Right, and in some cases you have Western blot as well. If the authors provide it, it is there. Right. So later on, as I said, we realized, okay, this is not enough. We are getting new EV subtypes, especially the microparticles or the microvesicles or the ectosomes or even the apoptotic blebs. So we started, okay, why don't we, because we already named it ExoCarta, we said, okay, let's think of something that unifies all the EV subtypes together, and then we can create a compendium that is going to include all the cargo that is present in EV subtypes as together, right? And that's why we created this microvesicles.org, which is Vesiclepedia database. When we did this, one main reason why I did this at that time was because of the time it took me to update ExoCarta. Because again, as I said, I was the only person who annotated it when we started. But later on, when I did a regular update, I was involving some people who are interested in annotations. Like, for example, I was collaborating with a group in US who was well known in lipidomics. And then Gavin Reid, and he now moved to Australia anyway. But at that time, he was in US. So we were collaborating with this group where they were one who were curating the lipid details. And then, because as you would know, when it comes to the standardization of names, if you look at gene names, you'll have gene ID, you have a gene symbol, and then the protein name is also standardized to an extent. And then if you look at lipids, it's not standardized as much at all, because you have all kind of lipid names, sometimes pretty much numbers in there. And I was not sure or not confident in terms of going and annotating this. So that's why I took help from these collaborators, and they did that for me in the later versions. So if this is the case, then I was thinking, how do we keep this data updated all through along? And one other thing that we thought of is involving the community. Like if we can ask people to submit their data when and then they can, and we can upload it into the database is what I thought at that time, right? So if you are try to say, if you're publishing a research article, let's say a cancer research article, or even a biogenesis article, a study that you're doing, it stops there, right? It stops 2 months into it. Then sometimes you can follow it up with follow-up studies, and then it may take another 10 years to finish that. But however, in terms of databases, there's no end to them, because you need to keep them updated and keep them on. So who is going to update this? Who is going to put the data? And when you talk about resources as well, there's not many people, many granting agencies who's going to support you to get any money for this, right? As of now, directly, I've not got a single dollar for any of these things that I'm doing. Because these are all part of the interest that I'm doing. I've got fellowships in a way to support my own salary, but not directly in terms of doing these databases and these tools. So we said, okay, why don't we involve the community? And that's what we did in 2012, where we reached out to the EV researchers at that time who were prominent in the field, and then asked them to contribute data to Vesiclepedia whenever they could. And then we provided certain formats for them to submit the data, and then we'll be able to upload that with not a lot of work, but very minimal work. We can upload it, is what I thought, right? But currently, even though we get a lot of data from many people nowadays, again, my job has changed from that time to now. Now I lead a group, and then because of that, I don't get a lot of time. And I have a bioinformatics person recently, but before that, it wasn't the case. So even that is not easy in terms of maintaining that, even though someone submits data to you, just getting that and collating that and doing it in a regular fashion is quite hard, right? But regardless, we were able to achieve what we wanted to do in terms of involving the community and get the database updated to an extent. Nowadays, we don't get a lot of submissions, I should say. We used to get a lot at one time, and we were not able to manage, but now we don't get that many. But however, this particular like effort in terms of involving the community did work, and then people were submitting data, and then we were able to upload that into Vesiclepedia. So currently, what we have in Vesiclepedia, that is one thing that we keep that updated often. And again, there's a lot of submitters, even in the audience. We may not respond immediately to that, but we have uploaded all those data to the current version of Vesiclepedia. And this moment, like say, Vesiclepedia, I think we updated the recent version in September last month, 2023. And it includes all EV subtypes that we know of, including exophores, including apoptotic blebs, even the non-EV subtypes, including exomeres and supermeres, are also cataloged into Vesiclepedia. And then now people can go and download the data. So looking at the statistics, again, it has kind of exploded. You can see there's more than 3,400 studies and these many entries in a way. When you look at exclusively in terms of the human proteome, how many protein entries uniquely are present in Vesiclepedia, there is close to 12,000 proteins that has been detected in Vesiclepedia, right? Again, uniquely I'm talking about. When you talk about the human proteome, it's like 20,000 proteins, and now 12,000 has been detected in EV subtypes, regardless of what EV subtype they are, right? Just says volume about what we know about the proteome at the moment. Plus also, one needs to be aware about the contamination that we may get from the studies, as well as the sample type that we are working with. So what I mean by sample type we are working with here is, let's say now we have this MySub standards where we talk about EV enriched proteins versus non-EV proteins. We talk about some ER proteins such as like GRPs and 96, as well as we talk about calnexin. But however, in some instances, you have to agree that these proteins also end up in EVs depending on the cellular state. If there is, like say, for example, unfolded protein response or some ER stress in particular cell, and then in such scenarios, these proteins are also ending up in EVs. It could be apoptotic blebs, or it could be even the small EVs. We don't know the precise mechanism, but one needs to be open about the fact that you cannot say a particular protein cannot be in EV. It depends on the context, right? So, but however, the MISA markers are indeed important in terms of the non-EV proteins as well. In many cases, they can help, but in some cases, you need to be open about that, saying that not all the time you can go with the logic that a protein may not be there in EVs, right? Depending on the context. So again, here we have covered almost like 56 species at the moment. We are trying to reach out to as much studies we can, bacteria, and then any species that we can update it. And we are welcoming anyone who wants to submit data to Vesiclepedia. However, I just have to acknowledge the fact that, again, we are only 2 people at the moment, me and the PhD student who's doing this. We may not be uploading the data directly, but we'll keep that on hold, and then we will upload it in the next version when we update it and then and there. Because if we cannot stop all the work and upload the data straight away, but however, we have a mechanism where we can do it once in 6 months and then have all those things collated together and then upload it, right? That's what we are going to do. And Sriram, who's also in the audience, is the PhD student who's going to do that. And then he will update you guys and email you guys if anything, and follow-up will be done when anything is needed in terms of uploading this data. So currently, we also have quantitative data incorporated into Vesiclepedia. Again, the quantitative data here is only available if a particular study provides that. So we cannot compare between studies as the logic would go. We can compare only within the study, provided the researchers have done that. So this is a snapshot of what you get from Vesiclepedia. You have the metadata, the experimental metadata, then which you can get the quantification of a EV cargo protein between the study. For example, if you're looking at whole cell lysate versus the EV, you can have that comparison. If you have 2 EV from a non-cancerous primary cell versus a cancer cell, then you can probably compare that as well, provided the study has done it. Plus, you can also look at the whole proteome or the RNA profile of a particular EV versus a whole cell lysate, and that can be available as a heat map as well. And then those experiments which has the EV quantification data is also listed via the browse as well as the query, right? So you also have the gene summary. If you go through one particular gene, similar to what you saw in ExoCarta, you will have the same gene summary here, and it will have some external links to other well-known resources like the Anthrogene, like the HGNC, the Human Genome Nomenclature Committee, like and then also the UniProt. Plus, you will also have more details about the samples from where you got this EV, the protein has been detected in which particular sample type from the EVs, right? And then you can also download all of these data, which is available in Vesiclepedia, either as a tab-limited file, or you can go to FunRich software and download this Vesiclepedia as a plugin, right? It's part of a plugin in Vesiclepedia. You can just click a button, it automatically updates the data, and then you can go and pretty much upload it inside your FunRich and do an analysis. So you can do comparison between your data set and FunRich and Vesiclepedia using FunRich. You can do a Venn diagram with that. Plus, you can also select some certain subsets and also do some gene enrichment analysis with that as well, right? So now, what is the advantage of this directly downloading from FunRich, you can have some filters there. You can filter it based on Homo sapiens, you can filter it based on an isolation method, you can filter it based on an EV subtype. All those options are available, and you can just go there and play with this data and see what you get out of this, right? So, and you also have this column in terms of this top 100 proteins, which is there in Vesiclepedia as well as ExoCarta. But Vesiclepedia, you have this. And this is a snapshot that I took from the recent Vesiclepedia page, right? You can see CD63 being the most identified protein, identified more than in 1,000 studies. You can see CD9, ALIX, TSG101, CD81. You can see all of these things are the most enriched proteins. You have also GAPDH, FLOT1, actin as like annexin, and then STCP. Then you have the Hitchcock proteins and all the others, right? So while this list is useful, this list is definitely useful when you're comparing against your particular study and saying that when I look at the top 100 identified proteins in EVs, and when I compare it to my studies, I identified these many proteins here, suggesting that my EV prep probably is relatively good, right, compared to what you get. But however, one needs to be aware, just because it's one of the top 100 proteins in EV databases, it doesn't mean it's an EV marker, right? So you need to be aware of that. It's the data that is present in Vesiclepedia or ExoCarta or any other resource that is available online is as good as the published article, right? So if we are collating or manually curating the data from there and putting it into Vesiclepedia, doesn't mean it's a gold standard, right? So you should be aware of that. And you should be critical of the data that you're getting from Vesiclepedia, ExoCarta, or any database resource that you have. So the filter is important. That's why you have to look at the species-based information. It's also important to look at based on isolation methods, and you can go to the metadata and look at it carefully so that you make your own judgments. And then the question that you're asking in terms of your research question is important for you to use such data in terms of however you want, right? But regardless, these particular top 100 proteins is definitely useful. And for example, if you identify CD9, CD63 more often, you know that these are bona fide enriched proteins on the EVs as we know now, right? So with that, I'll move on to FunRich. I'm just looking at time, so I'm doing well for time. So with FunRich again, functional enrichment analysis. The reason we developed this particular standalone tool, again, there are quite a few tools for enrichment analysis. We can talk about the Ingenuity pathway analysis, the IPA, that is commercial. When it comes to open source, you have a lot like the DavidDB, which is an online one which can help you. Plus also a lot of plugins that is available via Cytoscape. Like Panther is also one thing that can be done as well. So there's quite a few resources which are available that can do gene enrichment analysis, right? So why did we start this? We started this at that time, like pretty much in 2012, 2011. And then we finished this in 2014-ish in a way. So why we started on this is we wanted to create a gene enrichment tool that is intuitive for the biologists to use without the need for them to depend on a programmer or a bioinformatician, right? If anyone has a data, a proteomics data or an RNA data, they can just go and submit the data inside FunRich, do Venn diagrams for themselves, do heat maps for themselves. That is the primary intention for us to do this, right? Plus, then we also wanted to cater it for the EV audience to an extent, because again, that's why we created the Vesiclepedia plugin as well, so that people can play along with that. These are the two main reasons why we did this. An intuitive one, which kind of helps the biologist to do that. So you can access FunRich at this particular website, funrich.org. Again, it's free for download for anyone. It's based on Windows, and you can just download it and keep it in your laptop, and it's quite handy. It is not available in Mac. Quite a few people asked us to develop it for Mac. We were trying to make a web-based version, but later on, the effort was too much. And when you're making a web-based version, you would imagine when you're doing these kind of analysis on a real time, the speed has to be taken into account. The server has to be much more efficient. And then I thought it's not going to be sustainable for us to keep that ongoing, given the load that it will take for a server to run it all the time. So we just said, okay, we'll keep it pretty much as a standalone tool for the moment. And personally, we don't have any plans in terms of making this an online version in the future. But we have plans in terms of incorporating new features into FunRich and updating this as and when we go. But the good thing about FunRich is it is a template. You have a lot of options in it where you have certain options in a way where you can use this tool even after 4 or 5 years, even after 10 years, even after 20 years, without modifying too much to the tool in a way. So what I mean by that, I'll come to that later. But what can FunRich do? I will show a little bit, and then I'll come to that part as well. So now, again, this is one of the Venn diagrams that you can plot. You can just upload your proteomic or your RNA or any kind of omics data that you have, and then look at what is common between them or not, right? So sometimes what I do is it's real. Like sometimes when I'm dealing with students, like when I'm teaching in a way, when I have two lists of students and I want to see who are the common ones. For example, those who took test A versus test B, and I wanted to see who are the people who missed out or who are the people who commonly took both the test. Then I just throw them in FunRich and I try to get these lists there as well. It's quite easy rather than looking at in terms of what is rather than putting it in Excel and going through in terms of one by one, right? So now you can also do a three-way Venn diagram. In this, you can also do it in terms of the size of the data set. Here you can see every circle is of the same size, but however, here the circle is different based on the size of the data set. And then you can click on any of these numbers, and then you can download the entire protein list or the RNA list, the gene list, or just the names. You can also download the common ones. You can also say what is redundant between them is easily available in a particular clip as well. So you can also go for four-way Venn diagram, and the colors are all customizable. The text can be edited. The font is like whichever font you want, bold, regular. All this are publishable images, publication quality images, and you can just download it directly and put it into manuscripts. If you can look at some of our publications, you can see the Venn diagrams pretty much come from FunRich. We don't do much in those, so we just take it and then pretty much upload it. The heat maps also, we tend to use it from FunRich as well. But when you have more than 4 data sets, you can also do a pairwise step-based comparison, which can be done quite easily. You can just upload the data and then look at what is common between them. And again, this is available for many data sets, and you can say the colors and the text and all the data here is completely customizable. You can change anything you want. You can move anything in a way. So also functional enrichment analysis, you can do gene set enrichment analysis with the data that you have. So this is how FunRich looks, where you have the data that you're uploading on the left-hand side. You can say add data set here, and then you can upload the data. There is videos available on YouTube as to how you can work with FunRich. You can go to the help page in FunRich, and then you can look at the videos. And it's quite intuitive. It's not anything very hard to use. If you use Cytoscape, I'm sure people who are dealing with a lot of interaction network data, Cytoscape is the best software out there to do that in terms of there's a community working on Cytoscape. A lot of intelligent investigators or including a lot of plugins inside that. But however, you need to also accept the fact that the more and more options you have, it becomes very, very not so user-friendly, right? And it becomes very hard to navigate things, and it's tricky to keep a software user-friendly with providing a lot of options. But in this case, you can see that it's very user-friendly and very intuitive as well, because there's not many options here. It's very few. And then we are catering for what we are trying to do in terms of gene set enrichment analysis, as well as this Vesiclepedia plugin that you have here, right? So then you can also choose the database that you're working with. Here you can see there is a FunRich standalone inbuilt database. Then you also have the UniProt. Then you also have the custom, you have the Reactome, and also you have the gene ontology as well, right? So all those database options are available, and you can choose whichever you want. The beauty about this part is, when I said the software is sustainable even after 10 years, is the database. So the database, when you go to UniProt, it's one click and you can update the database today, right? And you can do the same thing after 10 years, provided the script has not been changed from UniProt in a way, and we can update that. But if they maintain then the current way, the software is going to go look at the recent database in UniProt, download it, and then create an updated database for you on the fly. Plus, you can also upload your custom database in terms of whatever features you're looking for, and you can annotate it in that way. And when you do gene set enrichment analysis, you can do it in two ways. One is you're comparing between two data sets. Let's say one EV versus other EV dataset. One could be a much more metastatic cancer cell-derived EV. One is a non-metastatic one, and you're looking at what could be the metastatic genes, right? In other way, you can also compare it against a background database. The background database here is the human proteome or anything else, and then you're comparing against that and trying to find if any of the proteins that are identified here, is there any pattern of a particular set of genes that is enriched? Is there a class of gene that is enriched? For example, in this case, you're looking at lysosomes, a cellular component. This particular analysis is saying that the lysosomal genes are enriched and the p-value is less than 0.01, right? And you can see the threshold is the red p-value here. And the percentage of genes that is identified in your data set is given here by the numbers as well as the bar. But the p-value for this particular thing is less than 0.01, and you have that here, right? And you can see each of those p-values are denoted in this. And you also have the column called exosomes here. And recently, this also has extracellular vesicles if you go into UniProt and then download the data, right? So I just want to take some more time here. So you can see this particular column, the Venn diagram. You can click on this and you can get the Venn diagram. You can edit the color and the fonts. And there's various chart types you can get. And that's what you're seeing here, where you can do the same thing in pie charts. You can also do a column chart in this, where you can have the p-value represented side by side along with your percentage plot as well. Where is the statistics coming from, right? So the statistics is pretty much, we are using the hypergeometric test. We also have the false discovery rate correction at the FDR, the multiple testing in this case, coming from Bonferroni, the pH method, and also the Q-value, which is the Story and the Tibshirani methods. So these all are the background statistics that is happening when we are doing the gene set enrichment analysis. But you have the option to choose any of them. If you go to the option menu in this one, you can see this FR button here. You can click on these options and you can change many of the parameters that you want. You can change the way the p-value is calculated. You can also report in terms of, I want the sorting based on p-value, or I want the sorting based on percentage. It's up to you. Or you can also choose in terms of in terms of how you are normalizing against the database, because most of the softwares won't allow you that option. So what I mean by normalize is, let's take an example of a species. Let's take even the let's take a bacteria which has not been studied well, right? But now you isolated the EVs from that bacteria, did a proteomic analysis, and then you ended up finding somewhere like 500 odd proteins in that. But when you look at the whole proteome of that bacteria, you have close to, let's say, 800 odd proteins or 1000 odd proteins. But when you look at the annotations for those proteins, 10,000 of those 1000 proteins, only, let's say, 50 to 100 of them have annotations. When I say annotations, these are metadata for a particular protein that says, okay, this protein is involved in iron metabolism, or this protein is involved in replication. So those kind of gene ontology annotations, some of the proteins have, but many of the proteins don't have those annotations, right? So in that scenario, how do we normalize this? So you're comparing your EV data set, you identified 500 proteins, and then you're asking the question, among these 500 proteins, is there a certain class of protein that is enriched? Is there iron metabolism proteins enriched in my EVs compared to the database, right? But if you have a database that has less annotations, are you going to compare the whole 1000 or only those which has annotations? So those kind of questions, there's no options which are available in many of the softwares. So they just blindly do it. And then you have to understand these options are available. And that's why this is what is given to the users in FunRich. But the default one is where we are taking only those genes that has annotations. If only 500 genes have annotations, you're only comparing against the 500 and not everything, right? That's how you do the gene set enrichment analysis, and then you use the hypergeometric test to identify the p-value there. So again, you can also compare between two data sets, right? As opposed to comparing against the database, now you're comparing against two data sets. Here, normal EVs versus patient-derived plasma EVs versus normal plasma EVs. And then you're looking at the percentage between them. And of course, you can calculate the p-values and see what is the significant ones you're getting from this. You can also do enrichment depletion curve, which is compared against each other or against the database. In this case, you're looking at pathways, the biological pathways that are present in, that are enriched in your EVs, are depleted in your EVs, which is in the red, and enriched ones are which are in your blue, right? So you can see the glycerol 3-phosphate shuttle proteins are enriched in your EVs compared to your background database or a particular data set. So this gives you more information, and similar to any gene enrichment analysis software, this kind of gives you that clue. Plus, you also have interaction network analysis in FunRich, but honestly, I would not recommend people to use this. It's quite good in a way, but however, there are quite a few amazing network analysis softwares out there which are coming from Cytoscape, which are developed daily, and quite a few plugins are automatically added to that. So I would suggest people use those softwares for that. But when it comes to gene set enrichment analysis, FunRich is pretty good. With that, I will probably stop here. But in this case, you have this Vesiclepedia plugin here. So you click on that, you automatically can download the Vesiclepedia data, and then you can upload your data sets and then compare against them. You can only look for certain data sets and then look for it. You can export all this analysis in an Excel sheet from here, and then you can create your own figures outside as well. There's no need for you guys to create use the figure that FunRich provides. You can also download it from here. Plus, you also have the heat map option, which kind of allows you to create a heat map for your data set. You have to provide quantification for that, or you can just upload the entire data or you can only upload certain lists, like say 10 or 20 genes, and get the heat map quite easily as well, right? So these are all the options which are available. And what we did in terms of FunRich at one point of a time is we went to, you have this column called request features, right? So we went to the community and asked them, what are the features? This is the software that we created as a template. What are the features that you guys want us to incorporate into FunRich, right? And we got quite a few suggestions. And then we started working on those which had a lot of votes. So it's a voting system. Whichever is the most popular feature that has been requested by the users, we started incorporating them into FunRich. And the options are still there. We incorporated almost 80% of the features that has been requested by the users, right? Except the Mac and the online version, we haven't done that. But other ones, we have incorporated one, and then it's pretty much there. And if anyone wants any additional features in FunRich, you can go there and request the feature, and then it automatically sends an email to us, and then we can look at it, and those which have been ranked, we will try to incorporate that into FunRich. With that, I'll stop here, and I'm happy to take any questions. Thank you guys for your attention.
Great, thanks, Suresh. That was a great talk. I'm sure a lot of people are already familiar with ExoCarta and Vesiclepedia and FunRich, but definitely a great overview, and nice to see that Vesiclepedia was updated. I think you touched on a couple of really interesting points about Vesiclepedia. So for me, like, using it as a reference and comparing to your own results is obviously the main question. It's like, how good did I do, essentially, compared to what's kind of been uploaded by everybody else? But it's not without its limitations, because it's only as good as the data that was generated in those particular instances. And I remember in the previous version of Vesiclepedia, albumin was definitely in that top 10 or top 20 list of most commonly identified EV proteins. But as we know, it's an abundant plasma protein, so always to be interpreted with caution. I guess, like, from a metadata point of view, it's so valuable to have that information. Like, what you've created is such a valuable resource, and the information that you could extract from that with regard to how things have progressed even over the last few years with regard to isolation techniques, improvements in proteomic or microRNA analyses, all of these things that you could probably publish many papers on if you had the time and energy to extract that information. And it's kind of similar in a way to what they're trying to do with EV Track, is to just get people to report on how they're doing things and what they're doing. But you've got this added element of, you actually have the data sets that have been uploaded. So what, like, do you have a vision for what this could maybe turn into? You kind of said you have some ideas for Vesiclepedia in the future, but do you have any ideas for actually collating some of that information and publishing like an update on where we were like 5 years ago and where we are now, or something of that nature?
Yeah, that's a good question, Nat. So we have spoken about this quite often, not only me, but with other groups as well. Again, in the conferences, we talk about it, and there was a time where not only these kind of comparisons, but also let's say you have this resource, you've collected a lot of data in that. Can we use it to predict something? For example, what are the protein features that help you in terms of sorting the cargo into EVs, right? So does a size matter? Like, is there certain motifs in the proteins which could help them? So like a lot of questions which could be used to mine this data. But I have reviewed a few papers recently, and there's quite a few which are coming out where they have these kind of concepts, and they've used ExoCarta and Vesiclepedia for that. For in our hands, when you're comparing between before 5 years and now, we do a little bit of those analysis in the publication as well, saying that what has improved, what have people studied. For example, 10 years ago, there was probably one isolation method. Now you can see most of the studies use a combination of isolation method in anything that you touch, right? So it could be ultrafiltration, which is not precisely a isolation method, but that is also always combined with an isolation method. In a way, you do OptiPrep with the ultracentrifugation in a way, or a size exclusion. So these kind of statistics helps the field as well in terms of seeing. And when it comes to plasma studies, what is the most predominantly used ones? But you see these kind of statistics also coming from myself papers as well. Like some of the standard papers we see, we get these data as well. Yes, there are plans for us to do these kind of analysis, but however, I think at the moment we are more interested in terms of looking at mining these data to identify if we can understand more about the biology of the biogenesis. We can understand more about how the cargoes are sorted. So that's what Sriram's project is also about, and that's what he's working on at the moment. Yeah.
Okay, yeah, true. Because obviously we know that not all EVs are created equal either. So what you detect in one EV from one cell type or one biofluid may not necessarily be in EVs from other cell types or biofluids. So yeah, a lot to think about there. I will go to the chat box now. So Archana has a question about how to submit the mass spectrometry data of biological fluid EVs to Vesiclepedia. Which I think you kind of said, but just to be clear, so if someone wanted to upload the mass spec data, how would you do that?
So again, Sriram has answered that to an extent, but what happens there is, like, say you have a tab called contribute to Vesiclepedia. In that case, you can go there, and we have provided an Excel template, right? Excel sheet as a template. You can just populate the data. So what we are looking here is metadata in terms of experiments. Same like what Ashley mentioned, Nat mentioned. So we have certain parameters in terms of what is isolation method, what did you do, and those reporting standards, plus your proteins which are identified by mass spec and Western. You can just populate it and then either upload it online. There is also a login which allows you to upload, or in a way you can just send it to Sriram and me. And again, Sriram will follow it up, right? So I may not be the right person in terms of replying to that straight away, but Sriram will follow it up, and he will upload the data once in 6 months, as I told you, in a way, right? But also there's a provision to keep your data private, which means like, again, for publication now, there's a mandate that you have to submit the data in terms of the raw data, as well as you can also submit your process data as well. So in that scenario, you can just give it as a private option to us, and then we'll not upload it in Vesiclepedia. And we also have an option in terms of you can upload it and keep it as private and not allow people access it. But once it's published, you can make it public, right? These are the two options which you can easily do.
Okay, great. Thank you. And yeah, I see Shriram's in the chat box also giving all the replies. It's going to be quite busy, I'm sure. Okay, so we do have another question from Chi-Yong. So I don't know if you want to unmute yourself and ask the question yourself. You're welcome to. Otherwise, I'm happy to do it. Okay, so it says, thank you for the wonderful presentation. May I know, is it possible to select only the top 20 proteins or microRNA for the functional analysis, because the low abundance for the left proteins might cause no significance for EV functionality? Hope that makes sense. So is it possible to just select the top 20 abundant proteins and microRNA for FunRich?
Yeah, I'm not sure I understand the later part of the question. The 20 proteins, yes, you can. You can download the top 20 proteins or top 20 microRNA, and then go and do an analysis, a functional analysis with that in any of the softwares, including FunRich. But that can be done in two ways. One is you can download the top 100 proteins, and then you can sort it by that directly, or you can go to FunRich and download the top 100 proteins from there as well, right? And the other option, in terms of, if you look at the second question, low abundance for left proteins might cause no significance. I'm not sure I understand the question, so I'm sorry. If you want to unmute and you can ask me the question again, if not, it's okay.
Yeah. Oh, sorry. Hi, Prof. Can you hear me?
Yes.
Yep.
Yes. How do I ask? Because we are new in bioinformatics, so I would like to ask: is it possible for us, for microRNA, like for NGS, we might get only one hit in our EV's microRNA sequencing. So can we remove those that only one hit in that maybe 10 power of 20 of EV's particles from that analysis, then to further our GO or KGG functional analysis?
Again, not sure whether I get your question, but what you're saying is if there is some data that is of low quality, can we remove that from that and do analysis with that? Is that what you're asking?
Yeah, something like that. But the main concern is because of those low abundance. For example, I got 300 of proteins from LCMS, the EV proteins from LCMS, but then the last 10 proteins which has very low abundance, and I feel like it might not cause any significant functionality for my EV set. So can I remove that to further my functional analysis? Is it applicable or?
I think if you're doing functional analysis, I would still do that with those proteins, because even though they're low abundant, sometimes you cannot say they are not functional in every case. It depends on the context. For example, let's say now we have a project which we look on cachexia, and then we in this case we are looking at EVs from cancer cells, which can cause, let's say, muscle atrophy or lipolysis. But these are low abundant proteins which we think are functional. And then when we do functional analysis with them, meaning when you do follow-up gene molecular biology, as well as when you do follow-up knockdown studies, we can see some of the low abundant proteins are very functional, right? So I would say when you do gene set enrichment analysis, you shouldn't discriminate like that. I would say I would still include everything. But however, another way for you to look at it is, like, say, example, you're looking at a normal versus a cancer, and then you're looking at certain proteins which are highly enriched in one versus other. You can just look at that subset alone. But removing something, I wouldn't suggest that for any functional enrichment analysis. I would still keep that in a way.
Then how about for microRNA? Because we know that microRNA is very low. Some of them say microRNA is very low abundance in EV, and maybe only 10 EVs have they only carry one microRNA. So how if for my NGS and sequencing, I found that there's only one hit for this microRNA? Then can I remove that one? Because from NGS, we need at least 20 nanogram of total RNA for the NGS analysis, the library preparation. Then, because only one hit means that only one hit of microRNA for that total RNA I got from that so many of EVs. I'm not sure.
Yeah, but if you're asking in terms of the quality of your data, right? So if your question is on the quality of your EV data, let's say if you do proteomics or even NGS, and you see the counts are less for something, and your counts in terms of your peptide identifications or your spectra identifications in terms of the proteomics are less. Again, there are several ways for you to tackle that. You can probably do let's say in terms of a false discovery rate, and see what is the false discovery rate of your data. Even though if you get one peptide, two peptide identifications, gone are the days where we throw them away. I would say still include them, provided you have a high sensitivity for your instrument, and then the resolution is also high, and then still you're able to get it. But in case of NGS, if your counts are low, I wouldn't include them, right? So there, again, the pipelines, there are a lot of pipelines which you have for you to throw off these data when you process the raw files, and then you go with your reads, and then you normalize them, then probably there it takes care of itself. But you manually deleting something in your mass spec file after it has passed the threshold, I wouldn't suggest. But in NGS, I can understand if the counts are not there, you shouldn't be in the list. So I'm not sure whether it's still I got your question. I think this is what your question, but maybe you can correct me.
Okay. I think we might just, in the interest of other people having a chance to ask questions, we might just move on to the next one. Yeah, perhaps you could reach out if you had any further questions about that. So if has a question, would you like to unmute yourself and ask your question? Otherwise, it's in the chat box, and it says, how would you explain detecting exosome proteins by Western blot, but then when doing LCMS, we cannot find them using UniProt? I think it's probably a very common problem, but Suresh, do you have a response to that?
Yeah, so we all struggle with that. It's not you guys alone. So again, in a way, so there's several reasons for this, because you need to understand here the mass spec is very sensitive, very sensitive, and in a way, like you subjecting one load, like a bolus concentration of proteins, and then you're identifying a lot of proteins here. Your Western, again, it may not be as sensitive as the mass spec, as we know, but now you have to understand, provided the sample is the same, right? So that's why I always tell in the lab, when you are doing proteomics with the sample, I would also ask them to keep a bulk amount of protein in the freezer for the same sample, meaning you can run a Western of the same sample later on, right? So why, when you have some doubts of this, right, the same problem. Here the problems also in a way is, in sometimes you get that in Western, in mass spec, you won't pick it because some proteins are not able to ionize. Some proteins are hydrophobic, right? So let's say if you have a membrane protein, the membrane protein may not be as much identified in a mass spec analysis as compared to a Western blot, because these have a lot of hydrophobic amino acids in them, and they are not going to be ionized. For a peptide to ionize really well, provided you're using the basic ones in terms of trypsin in a way, and then you're ionizing them, you need to have a lot of basic amino acids like arginine, lysine, plus other basic amino acids, then can fly well, right? So that is one of the problem, where it cannot be ionized well, and by mass spec, then you're not going to pick it. By Western blot, you can pick it. Then you also have low abundant proteins, right? So you have a lot of low abundant proteins which you're not going to pick in mass spec, provided you are optimizing that methodology, right? So again, mass spec, you're doing that based on the dynamic range, and then you have the top proteins which are identifying most often, but the low abundant proteins are not going to be identified unless and until you enrich for them. There are various techniques you can do to identify the low abundant proteins, and also set up the mass spec in that way, where you can exclude the ions that has already been identified, and only then sequence something that has never been identified, right? And you have certain tips that you can do, certain tricks that you can do to identify them again. And then the antibodies as well, right? Sometimes the antibodies can cross-react, and then when you have the antibodies in terms of epitope, right? We also see the opposite more often, as you can see. You do a Western, and then you only detect one band, but when you do a mass spec, you have so many of them in various locations, right? Because you have various isoforms, you have various proteolytic isoforms in terms of cleavage as well, but your Western may not pick it because your epitope is not recognizing that. But in your mass spec, it can detect in various ways as well. And then also there is this problem of common domains, I would say. Let's say there's common peptides, which can be found in many proteins. For example, if you have a conserved domain, let's say a PDZ domain, and that particular peptide is found in some proteins, you need to be aware of that as well. And then if your mass spec list pulls that out, you need to be careful in terms of how you're dealing. So the processing of your proteomics data is also important in terms of taking care of all these things. It depends on the context. So that's why I'm saying. So if you are doing Western and your mass spec doesn't identify it, I'm not too worried. If you're doing the other way, then I'm worried, meaning your mass spec is identifying and your Western is not identifying that protein, then I'll be worried, meaning that's why you have to have the samples of what you're used on the same day in your fridge. Now go back and then check, can I pick the same protein where it's enriched several folds in mass spec, but now I can't see that in Western. So now use the same sample. And if you're able to find that, then you know there's something reproducibility issue in a way.
Yes, so maybe if it doesn't work in Western and you detect in mass spec, it might be time for a new antibody. Also, if you aren't detecting in mass spec, it doesn't mean obviously it's not there. I think you can set up the mass spectrometer in so many ways with so many different methods. I found that in my proteomics results, I was able to detect things by a targeted method in proteomics, whereas I was unable to detect it in like a global proteomics method. So it depends on so many different things. So yeah, it depends on the context. If you're happy to stay for a few minutes longer, Suresh, we've got another question in the chat box. So Vincent, did you want to unmute yourself and ask your question? Otherwise, I will ask it for you. So it says, thanks for the presentation. While extracting small EVs from blood plasma, there is likely to be some lipoprotein co-isolated. Is it possible to use the tools presented to sort the proteins detected with mass spec and to separate those likely coming from small EVs and those carried by lipoproteins?
So is it possible to use the tools presented to sort? Yeah, it is possible. Like say, if you have, let's say, if you have the proteins that are presented in your lipoproteins, right, which we know of some proteins, for example, you have this apolipoprotein A1, apolipoprotein A2, and those kind of list, you can have it, and then you can compare it against them and see how much of that is present in each of the samples, right? And a lot of group has done that already, where they can say by the abundance that you find in your mass spec or your Western, you can just show that, okay, these proteins are depleted. And you can do the same strategy as well, where you can have in FunRich a list of these proteins, and you can call them lipoprotein contaminants, and then you can upload that and you can compare against your data set. And then you can even quantify between them, like see how much is present in here versus there. But one drawback of all these analyses is, like how Natalie pointed out before, albumin was one of the top 100 proteins, and then it is one of the plasma, highly enriched plasma protein in a way. Plus also the same applies to anything, like even I said this again, right? These proteins are definitely present in lipoproteins. Albumin is definitely a highly abundant protein in plasma. There's no need to validate that, right? But however, how confident are we to say that albumin is never present in EVs, inside EVs, right? Can anyone say that for sure? I'm not too sure, because when you even think about endocytosis, right? When you think about endocytosis, again, a portion of what is attached to the membrane is going inside the cell, and then this becomes a small EV, like exosomes, and then it gets shunted out again back, right? Even if you think of the classical mechanism, the classically secreted pathway, where you have the signal peptide, it gets cleaved off, and then these proteins get shunted out of the cell, right? They are in the extracellular space, and by endocytosis that can come inside an EV as well, right? And even there could be mechanisms by which some of these proteins can be packaged into EVs within the cell, which we don't understand, right? Which we do not know at the moment. So you cannot rule out that a protein which are known to be a contaminant is always going to be a contaminant just because they are detected in the EVs. But however, to your question, yes, you can do that. But however, you need to have caution that just because they are there, you cannot say, oh, they are not an EV protein. So you need to have follow-up validation, like maybe lysing them and then looking whether they are present inside them, even tracking them, and various other methods can be done to see whether they're inside or outside, and then make sure they're packaging inside in a way.
Perfect. All right, I think just one last quick question, and it's about Vesiclepedia. It doesn't at the moment contain any plant EVs other than citrus. So is there any update in the future when we could expect plant EVs may be included in Vesiclepedia?
Yes, definitely. We are including as much as possible in a way. Plant, again, as you would know by now, there are not many studies out there on plants, but again, the field is evolving. I can see more people interested in plant per se, and we are targeting certain sections now and annotating in the next phases. For example, plant is one of them. We did target bacterial EVs to an extent this time, so then we can add more as well. So likewise, we'll have phases where we'll target certain EV subtypes or from the sources, and plant is one of them, definitely. Sriram, are you listening?
Oh, Shahram, I was going to say, so please maybe don't email Shahram with every question or upload or anything like that. But yeah, certainly you two will probably see an influx of questions after this is published online. Look, with that, I think in the interest of time, I'll just say thank you very much, Suresh, for presenting today. It was excellent as always. Maimona and Amir, thanks for being here as well. Thank you everyone for attending. Maimona, did you want to say anything just as we're finishing up?
Just like to thank Prof. Suresh, and yeah, we're expecting a lot of questions. I'm receiving questions to our email now. Yeah, so yeah, thank you so much for your time and all the knowledge.
Thanks, everyone. Thank you.
Thank you. Thanks, everyone.